Hello everyone, this is your instructor commission, and this lecture would focus on the second part of our lecture on prokaryotic classification. We will concentrate on bacterial classification, bacteria that are gram-positive, and use the criteria of their G+C ratio as the main characteristic of grouping them. So the G+C ratio refers to the ratio of the sum of guanine and cytosine compared to that of the entire nucleotide together times 100%. So it's G+C, guanine, divided by the sum of adenine plus thymine plus guanine and cytosine. And then all this is multiplied by 100%. Now this ratio, based on the DNA sequences of the microorganisms, can be used to group gram-positive bacteria into either low GC content or high GC content. For this lecture, we will just concentrate on those that have the low GC content. So the phylum Firmicutes and the phylum Tenericutes. So for the phylum Firmicutes, we have members such as Clostridia, Bacilli groups, Cochlea groups that exist. For Clostridium, Clostridium is a gram-positive rod bacteria. They are obligate anaerobes, so they need environments that are devoid of oxygen. Oxygen is poisonous to them. But their very important feature is that they are endospore capable, so they can form endospores. And that makes them very important medically and industrially because endospore forming bacteria tend to be more pathogenic and are more difficult to deal with. So they can survive very harsh conditions in terms of treatment. So representatives will be Clostridium tetani, Perfringens, Botulinum, difficile, and we also have Venezuela species that are also of similar characteristics. But we'll just look at these examples and connect them to the disease that they cause, point out a few characteristics that makes them distinct. And so if you look at Clostridium tetani, it causes tetanus. In this picture, you see an advanced form of tetanus, which results in spasms, and this could lead to spinal fracture and death. And its results, this condition is called opisthotonus. So opisthotonus is this advanced form which could actually result in a spinal fracture. What you would notice with the morphology of the bacteria is that you see these enlarged areas. In this example, you see some that doesn't have that bulgy brackets-like structure, and then you see a number of them that do have that. Now those that have these bulginess, this bracket-like structure, are those that have formed spores. So the spore formations result in this bulginess in the morphology of the bacteria. Clostridium perfringens also would form spores, and you see the difference between those that do not have the spores and those that do have the spores. The spores will make them more pathogenic. In instances where there is an injury to someone that results in poor circulation, it creates an anaerobic environment that makes it very easy for these organisms to thrive. So perfringens can cause some of these effects on the tissues of individuals who have such conditions. That creates the right environment for them to spread. C. perfringens is also implicated in common food diarrhea. Then botulinum, Clostridium botulinum, is a spore-forming bacteria as well. As you can see, the spores in there. So these are having spores. Do not have spores. Okay, no spores here. Very similar to the other two members that we looked at. The difference is that this is a common bacteria that is found in the soil and in water bodies. Generally, if you should ingest the spores, it doesn't really do much harm to you. However, in anaerobic conditions, such as a sealed can or a sealed bottle like this, which we usually will use for preservation, what happens is that if the spores are present, the bacteria produces toxins. So the bacteria is able to release exotoxins, release toxins to the environment, which causes botulism. And botulism would result in a disruption of the transmission of nerve impulses and causes what we call flaccid paralysis. There's a reduced muscle tone. So the individual before having the paralysis and after. It could become very serious. It could lead to respiratory or cardiac failure and even death. Then there is Clostridium difficile. Clostridium difficile occurs in the gut as a normal microbiota. However, when someone receives heavy antibiotic treatment, especially a broad spectrum treatment, what it does is that it alters the composition of the gut microbiome and the diversity in there. And once an environment like that is created, they are able to cause infections such as colitis, the inflammation of the colon, the walls of the colon. And that may result in diarrhea, passing of blood in stools, and you can also see their endospores over there at the distended end as well. In addition to these groups that we just went through, there are some groups that are also low GC, gram-positive, but are described based on their shapes. So we have the Bacilli group and the Cochlea group. Now for the Bacilli group, you have Bacillus thuringiensis. Bacillus thuringiensis produces a toxin that is heavily used in the pest control industry. And so most of the pest control sprays and mixtures that are available on the market will contain Bt toxin. You should check your whatever control in terms of pest control resource you have in the home. Most likely you will have this component in there, the Bt toxin. Most likely it's going to be there. And it's been very effective in controlling pests. And then you have Bacillus anthracis. Bacillus anthracis causes anthrax. And anthrax will present, could be cutaneous, it could be respiratory, or it could be gastrointestinal. The respiratory type of anthrax has been weaponized or can be weaponized as a biological agent. And so there are instances where they are applied to letters or some other things that someone is going to touch or inhale, and it essentially might result in a person contracting the disease. Then you have Bacillus cereus, which generally wouldn't cause infections, but rarely would cause food poisoning in starchy foods such as rice. Then we also have Listeria monocytogenes. Listeria monocytogenes is a common contaminant of dairy products because they are resistant to cold. So usually we'll refrigerate the milk and the meat, and what they can do is that in such an environment, they are still able to thrive. They can grow at that temperature. When someone who is pregnant, if someone is pregnant, the person becomes infected with Listeria monocytogenes, it might result in stillbirth or birth defects in the fetus. And immunocompromised people may also suffer some effects from this particular bacteria. So I have some pictures of these bacteria under scanning electron microscope. And you can see here the toxin that is produced by Bacillus thuringiensis. That's the yellow structures that you see there. Then for Bacillus anthracis, as you can see the spore there, this is an example of a cutaneous anthrax. And this is also how Listeria monocytogenesis presents cellularly. There are other bacilli, and the last one we'll look at in terms of the other bacilli will be Lactobacillus species. They are generally found on mucosal surfaces, so surfaces that produce mucus. So in the trachea and the likes, you could find them there. Generally, they live in a very symbiotic environment. They produce lactic acid, as their name suggests, from carbohydrates or glucose fermentation. And this lactic acid is able to prevent other microorganisms from growing in an environment that they exist in. So they are very poor users of glucose because lactic acid fermentation doesn't produce a lot of ATP. However, because they produce lactic acid, the lactic acid impedes or inhibits the growth of other bacteria, and so they thrive in spite of their low efficiency at using glucose. And there's an example of what we call microbial antagonism. They antagonize other microorganisms and prevent them from growing. So essentially, we're able to use them in the food industry to produce buttermilk and sauerkraut and yogurt and the likes. So they are quite important in the food industry. The other group that we are looking at under the low GC are the cocci group. So we are still talking about the phylum Firmicutes. There are some cocci bacteria that belong to this group, and we have Streptococcus groups, and then we have the Enterococcus group. Now for the Streptococcus, they are known to be very pathogenic. They produce enzymes that will destroy phagocytic cells. Phagocytic cells generally are part of the immune system and they help fight infections. But these cells themselves are attacked and destroyed by enzymes that are produced by Streptococcus. The enzymes that they produce will also destroy connective tissue and fibrin. Fibrin is a clotting agent that we find in blood. And so they're able to destroy all these proteins that exist in our tissues. And so it leads to what we call a necrotizing fasciitis, a flesh-eating disease. And it appears very suddenly and spreads very rapidly. And the streptococcus that are implicated in these are known as the beta hemolytic streptococcus. Examples will be Streptococcus pyogenes and Streptococcus agalactiae. And then you have the non-hemolytic streptococci, such as strep mutans, that are also implicated in some of these diseases. We call them beta hemolytic streptococcus because they produce enzymes called hemolysin. So hemolysin or hemolysin. So hemolysin is able to digest the hemo content of blood, so there is complete digestion of it. When completely digested, we say beta. If it's incomplete, it's alpha, and if there's no digestion, it's gamma. Then you have the enterococcus. The enterococcus will live in environments that are low in oxygen, but rich in nutrients such as the intestine, the vagina, the oral cavity, and they are one of the leading causes of hospital or healthcare associated infections. They are commonly implicated in surgical and urinary tract infections as well. The last cocci that we'll look at will be Staphylococcus aureus. They are normal microbiota of the skin, in addition to Staph epidermidis. Then they are also able to survive environment with very high osmotic pressure, so environment that has a lot of salt in there. So now nostrils and all that, we have a lot of Staph aureus there because they're able to survive environment with high osmotic pressure, even up to 15% salt concentration. But they can cause opportunistic infections, so that they are implicated in diseases such as impetigo, where you have this contagious infection of the skin, very common in children and infants. This is how Staph aureus appears morphologically under micrograph. The phylum Tenericutes used to be part of the phylum Firmicutes, but so this is a new phylum that has been created. They compose of members that do not have a cell wall, so we describe them as pleomorphic. Because they have varying shapes, they don't have cell walls, so they have different shapes all the time. Remember, the cell wall gives the organism a distinct shape. So if it doesn't have a cell wall, it's just the cell membrane. So the outer cell membrane is the outermost layer of this particular organism. They are very small, ranging from 0.1 to 0.25 micrometer. They cannot be seen by a light microscope. They are facultative or obligate anaerobes, and they are very important in the medical field because they are implicated in pneumonia, common pneumonia, and urinary tract infections as well. And the micrograph, this is how they appear, and this is also the same depiction. What you will notice here is that you have these extensions here that are indicated by these arrows, and the extensions are extensions of the cells. So the cells will have these extensions that most probably help them in gliding or attaching to host cells. They are probably the smallest self-replicating microorganism that exists. A representative of this group is the Mycoplasma pneumonia, which is associated with a very mild form of pneumonia. In this picture, what you see is how it appears when it's grown on colonies. And the only way to observe that is under high magnification, very high magnification. They appear as fried egg colonies. So because they don't have a cell wall. So that's how they appear. So this brings us to the end of our part 2 lecture on prokaryotic classification.